Hey friends, this is Abhinata Party and welcome to my channel Bio with me. As you know, this is my first video and today I'm going to discuss the chapter Structural Organization of the Cell. Before that, let me tell you from which book I will give my explanations and for whom my explanations are relevant. So, I'll give my explanations from Nootan ISC Biology, and my explanations are relevant for both an ISC student, CBSC student, and also some competitive examination aspirants. Friends, why I decided to make this channel means uh, the major problem which I faced during my school days, as I am an ISC student, uh, not much content was available on internet. Neither the explanation for this good textbook, the Nootan textbook, is available on internet. So whenever I used to get stuck with some of doubts regarding the explanations provided in the Nootan textbook, I used to become hopeless. So in order to provide proper contents on internet for this Nootan explanation, I'm going to deliver the contents given in the Nootan ISC Biology. Friends, since this is my first video, I do not have much resources, so you might get some inconvenience. I'm sorry for that. So without talking much, let me start the chapter. And one more important thing, as I go on explaining the chapter, after each topic I will write the notes of some important points. Okay, so let's get started. So, the very first paragraph, the introduction paragraph, is the cell is the basic structural and functional unit of life. This concept, which is the part of cell theory, is proposed by a botanist, Matthias Schleiden, and a zoologist, Theodor Schwann, during the 19th century, some 20 years before the publication of Darwin's theory of evolution. This is the unifying basis for the modern day science of biology. Friends, as you know, this chapter, the structural organization of cell, deals with the cell and its constituent organelles. In this chapter, we'll study about what is a cell and what are its structural organelles. So, friends, as you know, cell is the basic structural and functional unit of life. What do you mean by that? And who gave that statement about cell? So this statement is one of the main postulates of the cell theory. This statement was given by M.J. Schleiden, Matthias Jacob Schleiden, and is a botanist, and Theodor Schwann is zoologist. And this statement means cell is a structural unit, means a smallest unit of structure, smallest unit of which an entire structure is constituted. Okay, for example, like a house. So what is a structural unit? Yeah, you guessed the right thing. It's the constituent bricks. The constituent bricks are the structural units of the house. Similarly, for an organism, for a living organism, these cells are the structural units. So the next thing is given, that is functional unit. So what do you mean by that functional unit? Functional unit means the function of an entire organism is due to the activity of its individual cells. You might understand this point in this way, as to perform a particular function we need energy. So the energy which we need is due to the respiration which is occurring in the each of cell. So this point was proposed by Matthias Schleiden and Theodor Schwann, and it is written somewhat 20 years before the Darwin's theory of evolution. So somewhat 20 years before, I will tell you the exact year, that is 1838. In 1838 the cell theory was proposed, and during that time only this point was added in the cell theory. Okay, so in the kingdoms Monera and Protista, the cell is complete organism, whereas in most fungi and in the animal and plant kingdoms, the organism is a highly complex arrangement of up to trillions of cells. So before that, let me tell you, Robert Whittaker has classified all the living organisms into 5 classes, and that is known as the 5 kingdom classification. They are classified as Monera, Protista, fungi, plantae, and animalia. Monera Monera is the kingdom of unicellular prokaryotes. Protista is the kingdom of unicellular eukaryotes. Fungi is the kingdom of multicellular saprophytes. Plantae is the kingdom of multicellular autotrophs, and Animalia is the kingdom of multicellular heterotrophs. So, once we come from Monera and Protista, they are unicellular, and this. Plantae and Animalia, they are multicellular. So this paragraph deals with the organism which are unicellular. For them, a single cell is the entire organism, and for organism, and there are other organisms also which are composed of many cells, and they are known as multicellular. Okay, then for example, here one example is given. The human brain alone consists of billions of cells. Billions of cells, I can't exactly remember. I think it is somewhat 1350 billion neurons. Yeah, so human brain is itself consists of billions of cells. So this means that there are organisms which are made of billions or trillions of cells. On the other hand, there are some other animals which are made of single cell. Then, cells and their activities are so vital to an understanding of life that the cell doctrine has become a central organizing principle in the field of biology. Yeah, so cell theory or the study of cell could be called as the central organizing principle or the unifying basis. Do you know why? Because after the study of cell, various branches of biology could be studied in a proper order, in an organized form. And what is science? Science is actually the systematic study. So the systematic study of biology could be done after the study of cell. Do you know how it is possible? Yeah, after the molecular constituents of the cell are studied, then molecular biology is studied separately. After biotechnology is studied separately. So after the cell, about the structural details of the cell came into existence, we got the explanations of other branches of biology. So in this way, cell is the central organizing principle of biology. Okay, now let us note the relevant things from this topic. In this way you will revise also. Okay, so the chapter name is so we are beginning here, structural organization of cell. Structural organization of cell. Friends, here, yeah, so from the very introductory paragraph, what is cell? Cell, what is cell? Structural and functional unit. Structural and functional unit of life. Okay, this point was given by M.J. Sladen. M.J. Sladen is botanist, and Theodore Schwann who is Theodore Schwann is zoologist. Yeah, so these are the points which we have collected. And one more thing, some organisms unicellular and some multicellular. Yeah, unicellular organisms are Monera and Protista, and multicellular organisms are fungi, plantae, and Animalia. And one more thing, study of cell is central organizing principle. Central organizing principle in biology. So these are the points which we have collected from the introduction part. Okay, so let us move further. Yeah, there's a great variety in size, form, structure, and function of cells. They range. So the next module, which next article which we are going to read is about the size and shape of the cell. So once let me introduce you with the subject. The size and shape of cell is not at all fixed. Okay, they depend on several factors, and there is a high range of size and shapes. Okay, so what might be the factor? The surface tension might be the factor. Surface tension of protoplasm, the viscosity of the protoplasm, the pressure exerted by the other cell, and the major one is the function itself, the function of the cell. That might be a factor for the shape of the cell. Okay, so let me read what's given, then I will provide you the explanation. Okay, there is a great variety in size, form, structure, and function of cells. They range in size from several centimeters in diameter. Example, ostrich egg, the largest cell, which is about 17.6 millimeter in diameter. 176 millimeter in diameter, 17.6 centimeter in diameter. Okay, to 0.001 millimeter. Example, mycoplasmas and some minute bacteria. So mycoplasmas about 1 micrometer in diameter, and the nerve cells found in mammals. The nerve cells, perhaps the longest cells, may attain a length of more than a meter. Similarly, fibers of certain plant may attain length of several centimeters. So friends, as you know, the size and shape of cells is highly variable, and what we call here some data are given, which is the largest cell. The largest cell is ostrich egg, and its diameter is about 17.6 centimeter. And smallest cell, mycoplasmas, which are about 10 to the power minus 6 meter. And talking about the longest cell is neuron, and yeah, some this fibers of certain plants are also very long, which can range up to several meters. Okay, now we are going to something important, that is what decides the shape of the cell. The shape of the cell, here some shapes are given, like this is a shapes of bacterial cell. Don't think the bacteria are multicellular. Actually, these are the colonies of bacteria. So bacterial cells are given here, and this is amoeboid shape or amoeba cell, which has no regular shape. Actually, some of the pseudopodia are projecting out, and this is actually the plant cell, isolated plant cell, and there is a cell wall around the plant cell. This is diatom cell, and here RBC, which are biconcave in shape, and WBCs are given, and this is nerve cell, and this is muscle. When we'll study about the muscle, human physiology, the movement and locomotion, I will tell you in detail what are the structures of muscle. Okay, this is muscle cell, and it contains myosin and actin filaments. These are two proteins which are present in muscle for contraction, and this is plant fiber cell. Okay, moving further, yeah, the shape of the cells is also much variable. They may be of any form and may change their shapes according to their position and functional adaptations. See the relevant point: position and functional adaptation. They may change also their shape. The cells of unicellular forms such as diatoms, amoebae, leukocyte, and bacterials have many shapes. Okay, and those of multicellular organism show still further variation. Okay, unicellular have many shapes, but multicellular still show much more variation. Now they may be disc-like, polygonal, columnar, cuboid, thread-like, or even irregular, like amoeboid shape, like WBCs. They might be irregular. Now the surface tension, what are the factors? Let us mark the factors: the surface tension, the viscosity of the protoplasm, the mechanical action exerted by the adjoining cells, and the rigidity of the cell membrane also affect the function, affect the shape of the cell. So one point we have collected in the beginning, that is the position and functional adaptations. So there are 5 factors which decide the shape of the cell. Okay, the positional and functional adaptations, the surface tension, the viscosity of the protoplasm, the mechanical action exerted by the adjoining cells, the rigidity of the cell membrane, they decide the shape of the cell. For example, in the stem and veins of a leaf, which are elongated structures, the cells too are elongated, particularly in conducting cells. Okay, so now let me explain how these things affect. So surface tension and viscosity. Surface tension and viscosity are a measure of the attractive forces present within the protoplasm. So if there will be more surface tension, or if there will be more viscosity, this indicates there is more intermolecular attraction within the protoplasm, and if there will be more intermolecular attraction within the protoplasm, the size of the shape will be reduced. Okay, so in this way they are deciding the size. And the mechanical action exerted by the adjoining cells, yeah, of course, if there will be much more cells around a particular cell, they will exert some pressure on this cell, and this way the size might get reduced and the shape might get deformed. Okay, then the rigidity of the cell membrane, yeah, if the cell membrane is much rigid, the shape of the that will also limit, that is also limiting the extension of the protoplasm. In this way the size could not expand much. In this way, and one more thing, position and functional adaptations. The position and functional adaptations of the cell are also the major factor. Okay, the position and functional adaptation. For example, let me tell you, neuron. Can you see the shape of neuron? Let me bring to your highlight. Yeah, neuron is much more branched. Do you know why? It has to collect the impulses from all the sides. Moreover, here is a myelin sheath. Since it has to collect and conduct the impulses, that's why the neuron is much more branched, and here there is a myelin sheath so that it can insulate. Okay, so what I mean is, this its function is to collect the impulses, that's why it is much more branched. Similarly, you can think of a sperm cell. A sperm cell has a tail, and the tail of the sperm cell helps in floating, and its function is to float and reach the female ova. So in this way there is a tail is provided in a sperm cell. So what I mean is, the function of the cell also matters for its shape and size. Now what points we have collected from this very article is, this next article is about size and shape. Okay, so let me note it, and you will revise also. If you want, you can also note size and shape. Size and shape. So here we are going to note some details, like nowadays some details are also asked, like largest cell, smallest cell, then longest cell, then after, yeah, these things. So here we are going to note the size. Okay, so longest cell, largest cell is ostrich egg. Size is 17.6 centimeter. Yeah. Smallest cell is mycoplasmas, which belong to the PPLO, pleuropneumonia like organism. Mycoplasmas, yeah, mycoplasmas, the size range is 10 to the power minus 6 meter. And yeah, the longest cell, of course, the longest cell is neuron. Okay, several meters, or more than a meter, we can write more than a meter. So these are some details. A meter. Moreover, here we are getting what are the factors affecting the shape and size of a cell. Note should be very what we call it should be very condensed form, so actually no need to write this much details. Let me, since I'm beginner. Okay, shape and size. Yeah, so factors affecting shape and size. So the factors are function and position and surface tension and viscosity of protoplasm, And the next thing, surface tension and viscosity of protoplasm. Then after, pressure exerted by other cells, other adjacent cells. Yeah, and something else, that is mm, yeah, limiting membrane, rigidity of the cell membrane. Yeah, rigidity of the cell membrane. Cell membrane. So these are the points which I have collected from this very article. Okay, now moving further, cellular organization. What actually is cellular organization? How a cell is organized? In the 19th century, the cell was described as having a limiting outer membrane, an interior nucleus, and a large mass of cytoplasm surrounding the nucleus. However, finer techniques of probing the cell gradually brought details of its internal structure to light. Improved staining techniques enabled researchers to heighten the visibility of cellular structures selectively. And later on, so before that, sorry, I forgot to explain the meaning of the word probing. That might cause a problem for you. Probing actually means investigating. So let me write the word meaning: investigating. Okay, investigating. Probing means investigating. Yeah, so this investigating so initially the cell was thought to be just merely a mass of cytoplasm containing a nucleus and surrounded by a plasma membrane. But later on, as much more detailed study was done in field of cell, okay, cytology means the study of cell. Okay, so much more work was done. That means much more tools were provided, like electron microscope in 1931. And so we could probe, we could investigate the cell, and after investigating we could find some structures which resembled like the organs. Those structures resemble like organs of particular person's particular organism's body. How the organs are working separately, performing their own functions. Similarly, the cell, the different structures which could be identified, those structures were also performing separate functions. And those structures, after performing the functions, they were performing separately. Okay, so since they were performing the functions separately, this this was comparable and relatable with the functions performed by the organs, and thus they were given the name organelles. Okay, so we will later on discuss each organelle of a cell in detail. Okay, so let me read the thing out. In the 19th century, the cell was merely thought of as having a limiting outer membrane, an interior nucleus, and a large mass of cytoplasm surrounding the nucleus. However, finer techniques of probing means investigating the cell, gradually brought details of its internal structure to light. Improved staining techniques enabled researchers to heighten the visibility of cellular structures selectively. Afterwards, besides the discovery of electron microscope, there were some stains, some dyes improved. So what are stains? Let me tell you what are stains. Stains are actually some dyes which could like it could highlight a particular organelle, because they are accepted by only particular organelles, and organelle specific stains are also there which are accepted by certain organelles, and thus they highlight that particular organelle, and they make our examination of that particular organelle much more clear. Okay, so improved staining techniques enabled researchers to heighten the visibility of cellular structures. So they are making the visibility of cellular structures clear. Later, vital stains were employed to study living cells and many subcellular features. Subcellular features means within the cell some features are there. Those could be studied now. Many subcellular features were studied, including chromosome and nucleolus of the nucleus. Okay, so we could now peep into the nucleus. We could now see the chromosomes. We could now see the nucleolus, which are located in the nucleus. With the development of electron microscope, so the electron microscope was developed in the year 1931. So with the development of electron microscope in the 1931, the elaborate membrane architecture of the cytoplasm was much more completely resolved. Okay, so before that it was not resolved, but now after the better probing techniques, it could easily be resolved. Okay, and in the next page, let me bring to your light the next page. Yeah, of course, since I am new, my setup, try to adjust please. I'm sorry for the inconvenience. Yeah. Yeah, so in the next page there are some details of some plant and animal cell are given, and then after some lines are given with the structural and functional details of subcellular structures available. It became clear that the functions of the cell are accomplished by specialized structures comparable to the organs of the body. Consequently, these structures were called organelles. Consequently means as a result, these structures were called organelles, which provide regional separateness like the rooms of a house and permit specialization. Okay, so what specialization or compartmentalization means? They are providing specificity for a particular function. Like in a particular house, there are special rooms for particular functions, like for preparing the food there is kitchen, for sleeping there is bedroom, for eating the food there is dining room. So likewise, for particular functions in a house there are particular rooms, particular compartments, only specialized only for a particular function. Similarly, within the cell there were structures which were specialized for particular functions. That's why these were for providing compartmentalization, and these were named as organelles since they resemble the organs of the body. Okay, now one more thing, the form assumed by the organelle related to its function. Okay, so the organelle, the form which the organelle assumed was actually related to its function. What the function it performed, it was related to that. Now let me note what we have collected from this topic. So the topic which you have read now is cellular organization. So let me bring to your light. Yeah. So the topic is cellular organization. Yeah, so here we have read about what was the initial view of cell. Initial view of cell means initially the cell was thought to be, initially the cell was thought merely to be a mass of cytoplasm containing nucleus surrounded by cell membrane. Yeah, and later on, with improved probing techniques, like, what are the probing techniques? Like, what are the probing techniques? Like discovery of electron microscope, in 1931, and improved staining techniques. Techniques brought to light small structures which resembled organs and thus called were called organelles. They provide specialization or compartmentalization. Friends, so this was my first video, and hope you would send your feedbacks through comment so that I could improve, and later on I will try to develop much more infrastructure so that I could upload much more content. So here I pause today. Okay, thank you very much.